What I'd like to tell you about is how do we use genetic variation in concordance with epigenomic and transcriptional data sets to try to understand the mechanistic basis of genetic variants and how they act on human disease. So a lot of MPG is focused on generating these kinds of plots and then using them to gain insight into the function of genetic variation. So here I'm showing you the result of a genome-wide association study, or a GWAS. So what you do is you do a chi-square statistical test for every genetic variant in the genome, and then you ask, for each one of these genetic variants, what is its association with a phenotype of interest? In this particular case, we're looking at obesity, or body mass index, a measure of obesity. And what you can see is that there's a large number of genetic loci that are above genome-wide significance, which is at 10 to the minus 8 over here. Reigning supreme among all of them is the FTO locus, which was discovered in 2007, but its mechanistic basis remained unknown for almost a decade thereafter, until our collaboration with Melina Clausentzer actually dissected its function. So why do we need to worry about these regions? The reason is that they give us a mechanistic insight that something in there has something to do with disease. And therefore, we should be able to understand the disease mechanism, discover new target genes for therapeutic targeting, and ultimately enable personalized medicine. The challenge, however, is that when you look within these regions, you find that the vast majority of these loci are devoid of any protein-altering variants. In 93% of cases, you look under the hood and none of these genetic variants is in fact perturbing a protein. So that means that we're a little bit at a loss as to what is the target gene, which one of these genetic variants that are co-inherited in these loci is the causal one, and what cell type do they act in. We can't just look at the expression of the target gene and say that's where it's expressed. We have to look at the non-coding genome, and therefore it's very hard to find the pathways and the mechanisms. In this particular case with FTO, you basically have 89 common variants sitting in the first exon of the FTO gene, none of which is perturbing the gene directly. So even though a lot of work was focused on understanding the function of the FTO gene, it turns out that the true targets are sitting 600,000 nucleotides away and 1.2 million nucleotides away. At this resolution, it's the building next door. So it's really, really complicated to understand these functions. And the one that I'm going to be focusing on today is the epigenome. So what is the epigenome? The epigenome is the collection of DNA accessibility patterns, DNA methylation patterns, and histomodification patterns that modulate the genetic information and tell the cell what to turn on in whatever cell type it is. So as you all know, all of our cells in our body have more or less the same genome, the same genetic material, but they perform vastly different functions between neurons and, heart cells and muscle cells and immune cells and, your hair and your nails. They all perform vastly different functions. And that's because the epigenome is modulating that genomic information and using different parts for different cells. But the epigenome can also give us insights not just on how genetic variation acts in every cell type, but also how genetic variation varies from person to person. So we can also tell us how the phenotype is in fact affecting the molecular processes of the cell. Because the epigenome is governed not just by your genetic makeup, but also by your environment. The epigenome also helps integrate all kinds of additional information about your own biology, like your age, your sex, other societal factors, socioeconomic status, and so on and so forth, are in fact influencing the epigenome. And the epigenome itself is influencing a lot of other processes, such as gene transcription or, splicing or microRNAs or lincRNAs. And it is interacting with a lot of other behavioral factors and microbiome and, immune history of infections and so on and so forth. So I like to think of the epigenome as the great integrator because it sits in a very sweet spot between genetic variation and disease, and it tells us about all kinds of additional processes. So what the talk will focus about today is different steps of utilizing this epigenome to understand disease mechanism. First, how do we even map and characterize the circuitry of the non-coding genome? How do we then identify disease-relevant tissues, variants, and regulators for specific traits? How do we combine genetic, epigenetic, and transcriptional variation in the context of disease? How do we integrate information across multiple phenotypes using rich medical records in the context of genetic information and molecular information? How do we then use that information to dissect the function of regulatory regions at the single-cell level? So we're going to see how we can use single-cell RNA-seq and single-cell attack-seq and other methods to start understanding the cell-type specific functions within complex tissues. And then we're going to go back to that first slide and see how we can combine all of that information to uncover the function of individual genetic loci. And lastly, how do we scale that up in terms of throughput to test millions of elements in a single experiment? So first of all, how do we map the epigenome? As I mentioned earlier, the epigenome varies greatly from tissue to tissue. So what you need is a map of the human epigenome across many tissues and cell types. And this is exactly what the NIH has been focused on and many other organizations. So we were part of the ENCODE project, the Roadmap Epigenomics project, that basically sought to now systematically map the human epigenome across many different tissues. And in each of these adult, embryonic, and iPS-derived and primary cells, we basically mapped multiple histone modification patterns, multiple ways of measuring accessibility and DNA methylation, as well as what we like to think of as the output, perhaps, of the epigenome, which is RNA expression. And this is what these modifications look like. If you look at DNA accessibility, it basically tells you where the nucleosomes are positioned, and therefore what regions will be accessible to different transcriptional regulators to bind. DNA methylation is usually associated with repression of non-coding regions, and it is acting directly on the DNA to inhibit the binding of particular regulators. For histone modifications, these are by far the most complex. So as you know, DNA is wrapped twice around each nucleosome. Every nucleosome consists of 8 different histone proteins, and every one of these histone proteins has a long amino acid tail. And these long amino acid tails can basically undergo more than 100 different types of post-translational modifications. For example, 3-methyl groups on the 4th residue, which happens to be a lysine for histone H3, or H3K4 trimethylation, is a mark associated with promoter regions. This is a mark associated with enhancer regions, with transcribe regions, repressed regions, repetitive regions, heterochromatic regions, and so on and so forth. And we have basically developed an arsenal of techniques for combining these individual epigenomic marks, for mapping them, and then for combining them to infer the function, quote unquote, of the epigenome, or if you wish, the activity signatures of the epigenome. And we've defined these activity signatures for enhancers, promoters, transcribed regions, repressed regions, and so forth. So we've written computational programs like ChromaHMM to basically automatically learn the language of the epigenome by inferring what are the most frequent combinations, the most relevant combinations of these marks, learning both the combinations and where they occur across the genome, and then annotating them with different classes of functions. So what that allows us to do is now go from these very many different tracks of individual marks to a single summary track that basically is much more directly interpretable. And these are the colors that you see at the top. So you can see transcribed regions in green, repressed regions in gray, enhancer regions in orange, promoter regions in red, and so on and so forth. The advantage of doing this type of summarization is that you can now look at a single summary track for each cell type and tissue, and then observe how these tracks are changing from cell type to cell type and from tissue to tissue. You can see here in IMR90 lung fibroblasts, this gene, PAX5, is repressed, as we saw in the previous slide. None of these activating marks are there, and in fact you see this H3K27 trimethylation mark, which is associated with polycomb repression. And you see the same thing in most of the other cell types. This gene appears to be off all the time. But what's really great with this dynamic view of the epigenome is that you can see that in a subset of cell types and tissues, it turns on. And that's kind of exciting. So if I have genetic variants associated with each gene, I know what cell type to look in. The other thing it allows you to do is look at how dynamic are these different classes of marks. So promoter regions in red are much more stable across cell types. Even when the gene is not transcribed, you can see that the promoter region is like ready to go. Enhancer regions, by contrast, are much more cell type specific. You can see that they only turn on in a subset of the cell types. And the other thing that you can do is actually use that information for linking. So if I have a non-coding variant here, I can say, well, number one, where does this non-coding variant turn on? Where does the corresponding region appear to be active? And number two, what are the genes that correlate in its activity? And what you can see here is that this gene, PAX5, is in fact correlated with the activity here. So if I'm trying to understand the function of genetic variants in this region, I shouldn't be looking just at the nearby genes. I should also be looking far, far away. Everybody with me so far? So that's one way of linking genetic variants to their target genes. As I mentioned in the case of the FTO locus, the true target gene is actually 1 million nucleotides away. So you can actually use these correlation patterns to find the true target gene or genes. You can also use high-C information. This information about chromatin conformation capture measured genome-wide. And in fact, here I'm showing you the same locus, the FTO locus, where the genetic variant associated with obesity is sitting here. And you can see how it forms long-range interactions with RX3 and RX5 that are sitting 600,000 and 1.2 million nucleotides away. And in fact, it forms these interactions with a whole 2 megabase block on one side and another block on the other side. So these regions are looped together in 3 dimensions. And that means that any of these genes might actually be the true target of these locus. And that's the second type of evidence that we're using. We're using functional correlation in the activity patterns between enhancer regions and their target genes, and also physical folding of the genome in 3 dimensions. The third type of information actually comes from genetics itself. So you've heard throughout this series about EQTLs. These are expression quantitative trait loci. The idea here, instead of just doing a GWAS where you're looking at how genetic variation correlates with phenotype, you're now doing a quantitative trait analysis where your quantitative trait is expression. So you're looking at the expression level of nearby genes, and you're correlating that with genetic variation across individuals. And if you find a correlation between the two, that means that this regulatory region is somehow linked to this target gene. And this is the third type of link information, genetic linking. Everybody with me so far? That's all for going downstream. I have a genetic variant. I want to figure out what does it target. What about upstream? Who are the regulators that control this genetic variant? To do that, what we can do is in fact cluster these enhancer regions based on their activity patterns, and then look within clusters of coordinated activity for common regulatory motifs that are enriched within these clusters. And based on the identity of the regulators that we know are targeting these motifs, we can in fact start saying that perhaps, if I look at enhancers in liver, the most relevant motifs and corresponding regulators might be HNF4A and an HNF1B, which are hepatic nuclear factors, which makes a lot of sense for liver, or GATA3 and SPY1 for immune cells, and so on and so forth. So that allows us to now start predicting who are the upstream regulators controlling these variants. So if we put all that together, what the epigenomic information is giving us is, number one, the cell type of action of these genetic variants. Number two, the regulators upstream that may be controlling these enhancers and perturbed by genetic variants associated with a particular disease. And number three, the target genes based on expression correlation, physical proximity, and EQTL information. Everybody with me so far? So we basically gathered all that information and we've put it out together in this database called HaploReg for haplotype regulation that basically allows you to look at every genetic variant associated with a particular trait, and then all of the genetic variants that are co-inherited with that genetic variant, and what are the specific tissues and cell types where these variants might be acting, what are the specific motifs that are altered by these SNPs, by these single nucleotide polymorphisms, what are the proteins that are bound there, what are the accessible regions, the promoter regions, are these regions evolutionarily conserved, and also what are the target genes. So we've built this for many resources, including the Roadmap Epigenomics project. Right now, we're updating this database to also include more than 100 different cell types that have been recently profiled by the ENCODE project. So if you look, however, at the dimensionality of how are these cell types covered, these are the numbers of assays and these are the different cell types, you can see that this matrix is quite sparse. And we'd like to have rich epigenomic information for all of them. So we've developed techniques for imputing the missing data by exploiting the correlation structure of these data sets, both within a cell type, how different marks relate with each other, and across cell types, how related are different cell types to each other. And together, we're able to now start imputing epigenomic information and inferring both enhancer regions and promoter regions across hundreds of cell types. So stay tuned, this is coming to HaploReg soon. Given all this information, we'd like to now start focusing on GWAS. I basically told you how we can do that at the global level. Let's now go into specific traits and see what does the data tell us about these traits. So what does GWAS give us? It basically gives us regions of the genome in a completely unbiased and agnostic way, regions of the genome that are associated with traits and specific nucleotides within these regions that are driving these associations. But what's really cool is that if you look across different traits, these regions are in fact changing. There's a different set of genomic regions associated with height, with type 1 diabetes, blood pressure, cholesterol, and so on and so forth. And therefore, we can now start using these traits relative to each other to ask what are the epigenomic annotations that are the most enriched in each of these traits. So we're going to start overlaying epigenomic annotations of enhancer regions that are active in stem cells, enhancer regions active in immune cells, in heart, in liver. And then we're going to say, are they enriched in specific traits? And what we find is, in fact, this kind of diagonal that basically says genetic variants associated with height appear to act in stem cells because they are enriched in enhancers active in stem cells. Type 1 diabetes in immune cells, blood pressure in heart, cholesterol in liver, and so on and so forth. So it gives us an insight as to the cell types and tissues where the particular traits may be active. Who's with me on this one? Awesome. And of course, this is a tiny little subset of a huge matrix, which you can see here, of 58 different traits and more than 100 different cell types, where you can see here that indeed genetic variants associated with height do not enrich anywhere but embryonic stem cells. Immune traits, T cells and B cells. Blood pressure, only heart, the left ventricle of the heart specifically. Cholesterol, specifically liver. Fasting glucose-related traits, very, very specific enrichment for pancreatic islets, completely consistent with the type 2 diabetes biology. Inflammatory bowel disease, you see this broad enrichment for immune cells, given the inflammatory processes, and also for digestive tissues, which again makes a lot of sense. If you look at Alzheimer's disease, what you would expect to see is an enrichment in brain, because of course that is the tissue that is affected. But we don't see that. And the reason for that, which we're going to come back to when we talk about single cell, is that the brain samples that we have are bulk tissue rather than specific cell types within the tissue. And if you look at bulk brain, more than 60% of cells are in fact neurons, excitatory inhibitory neurons. Another big chunk is astrocytes and oligodendrocytes, and the microglia are only a tiny little subset. These are CD14 monocytes, similar to the macrophages that are circulating in your blood, but they're sitting, specifically within your brain. And indeed, when we look at where is the strongest enrichment for Alzheimer's disease, we find CD14 plus monocytes, suggesting in fact that the microglia may be the first root cause of Alzheimer's. That's where the genetic variants appear to be acting prior to any other changes. To look at that more directly, we collaborated with Li-Hui Tsai from the Picaro Institute to look at the epigenomic changes that are happening in the brain of a mouse. Mice are much more generous with their brains than humans figure, and therefore we can look at early progression of Alzheimer's and also late progression of Alzheimer's. And what we found is that there is an activation of immune processes and a repression of neuronal processes during the progression of Alzheimer's. But the immune changes are happening early on, and the neuronal changes are only happening later on, again suggesting that inflammation and these immune processes are in fact the causal component of Alzheimer's before the neurons are even affected. So we developed a series of computational methods to now start integrating epigenomic information and genetic information and linkage disequilibrium of the co-inheritance patterns of these genetic variants systematically in order to predict what are the relevant epigenomic annotations. So for Crohn's disease, the system automatically predicts immune processes as the most relevant annotation. For schizophrenia, it predicts central nervous system epigenomic annotations as the relevant annotation. And then using these as empirical priors, it now goes back in every locus and it says, among all of these gray points which are associated from GWAS, I'm going to prioritize only the red points, which are the ones that have both genetic evidence and epigenomic evidence consistent with this genome-wide empirical prior. Everybody with me so far? And then we can actually ask, are these gray points and these red points showing similar enrichments for evolutionary conservation or for EQTLs and digital genome footprints, high-resolution accessibility inferences? And the answer is that these red points that are prioritized by the system are much, much more highly enriched for these independent sources of information, giving us a hint that perhaps we are indeed prioritizing functional loci. But instead of just prioritizing SNPs and genes within specific loci, what we'd like to also do is prioritize entirely new loci. If you look at genome-wide association plots, if you look at genome-wide association plots, what you see is, which we call Manhattan plots, what you see is these very tall skyscrapers that are sort of standing out over the New York skyline. And then you also see these very tall, 80-story buildings, which are taller than the 40-story buildings, but not as tall as the skyscrapers. The question is there anything useful there? And the answer is, well, it kind of depends on your cohort size. The larger the cohort, the more likely you are to recognize whether these are real or not. But what if we use the epigenomic information to actually prioritize these sub-threshold variants in this twilight zone between 10 to the minus 4 and 10 to the minus 8? And the answer is that you can, in fact, train a machine learning model using the skyscrapers to recognize epigenomic annotations associated with disease, and then turn around and apply it in this twilight zone and prioritize new loci that are now predicted to be causal drivers by combining the genetic information and the epigenomic information. So are these real? A lot of people laughed at us when we basically said, hey, can you validate this thing that's 1,000 times less likely than the genome-wide significant ones? Oh, yes, we know, you haven't even validated those yet, but humor us. And they did. And what we found in collaboration with David Milan over at MGH is that if you go in zebrafish and you perturb these loci that are not genome-wide significant, but are predicted using this combination of genetic variation and epigenomic variation, that are predicted to change heart repolarization duration, and you change those target genes, indeed you find that the heart repolarization duration changes in zebrafish. And if you look at mouse phenotypes, when you knock down these genes or you knock out these genes, you find indeed that they lead to cardiac conduction and contractility phenotypes. And if you look at human, not just QT interval, but QRS interval, a different trait which is related, you indeed find that these loci are much more likely to be associated with that than other loci at the same twilight zone, but outside enhancers. So that suggests that even before the cohorts become large enough to capture these lower association loci, we may be able to find actionable target genes. Because when you go in and change these therapeutically, you indeed see a phenotypic change. Once you see that, you don't need GWAS anymore. You now know that you have a possible therapeutic target that is supported both by genetics and epigenomics. So everything I've told you so far has relied only on genetic variation across individuals. And we've used reference epigenomes as if everybody in this room had exactly the same epigenome for every one of their cell types. The answer is, of course, much more complex. So what we'd like to do is not only use reference epigenomes, but also start observing how the epigenome itself is changing from person to person, and start painting a much more rich path between genetic variation and disease. What tissue is it acting in? Whether it's changing, a particular genetic variant is changing enhancer activity. What are the genes affected? What are the intermediate phenotypes, and so on and so forth. And of course, I'm drawing this as a one-directional arrow going all the way to disease. But the problem is that as soon as we leave this genetic information space where we have causality, all of these intermediate phenotypes are no longer unidirectional. They're now bidirectional. Why? Because they may simply be a consequence of the disease. Any kind of change that I see in enhancer regions might be consequence rather than cause of the disease. Or it just might be correlation. Some environmental factor may be affecting both my enhancer activity and my disease status. So we're going to have to develop new techniques for overcoming this particular challenge. So to do this, we basically collaborated with the ROSMAP cohort led by David Bennett at Rush University, and also collaborated with Phil de Jager at the Harvard Medical School, who's now in Colombia, to look at epigenomic variation in the post-mortem brain of hundreds of individuals that David has followed for more than 10 years using cognitive evaluations and hundreds of phenotypic assays, whose brains were donated post-mortem. So now what we can ask is, do we see epigenomic changes in the brain that correlate with the Alzheimer's status of these individuals? And specifically, whether genetic information can allow us to predict this epigenomic information, and also transcriptional information gathered for the same individuals in the same region, and also how together we can in fact use this information to infer causality about how these loci are acting. So to look at the first question of, is the genome enabling us to predict the epigenome, we can basically look for methylation QTLs. These are quantitative trait loci, where the outcome is methylation level. And what we find is that across the genome, we can find 50,000 loci where the genetic information is in fact significantly predictive of the epigenomic information. In here, if I know your genotype, whether you have 0, 1, or 2 copies of the particular allele, I can actually pretty well predict your epigenome. And what's really cool here is that I can know your genetic information at birth, and I can predict your epigenome in your brain at 93 years of age. That's a big deal, because it gives us causality back. Because if we can predict the epigenome at birth, it's not just a consequence of the disease, it's not just a consequence of the environment or behavioral choices that this person made. It is truly driven by the genetic variation of that individual. So we're going to use that information now to do something very cool. Instead of doing GWAS, which goes from genetics all the way to disease directly, instead of doing methylation QTLs that basically go from the genetics to the methylation, and instead of doing MWAS, methylome-wide association studies that basically look at the correlation between methylation and disease, we're going to look at a combination of them. So what is the challenge with MWAS? Methylome-wide association studies are basically saying, hey, this methylation level here is correlated with disease, but it's a bidirectional arrow. You don't know about causality. If instead I do something that we call imputation MWAS or IMWAS, what we're going to basically be able to do is say, not methylation, but the genetic component of methylation. The methylation that I can predict at birth is correlated with disease. And if that's true, then I can have causality back. Is everybody with me? Cool. So the other thing with IMWAS is that it's much more well-powered, because instead of having a small number of individuals that donated their brain post-mortem, we can ask a much larger number of individuals to donate a blood sample. People are much more generous about their blood. And we can basically carry out that study in living, breathing individuals by basically measuring genotype and whether they're sick or not, and so on and so forth. So this is very broadly applicable. So using this idea, we are now going through and measuring genome-wide methylation probes that are associated with a phenotype based on their imputed methylation from their genetics. And what we can see is that here across chromosome 6, we find half a dozen regions that are significantly associated with disease based on their predicted methylation level. But the corresponding genetic variants are not individually significant. None of them rises to 10 to the minus 8 here. Instead, it is by combining multiple genetic variants together that we can predict the impact on the disease phenotype. And we can carry this out not just with methylation, but also with transcription. So there's been a lot of work on TWAS and PredictScan and other methods that basically go through this path. We can actually combine these two paths, go through methylation, through transcription, and so on and so forth, and any other intermediate phenotype, to basically start predicting both regulatory regions and genes that are associated with disease. When we do this across the genome, we basically find hundreds of genetic loci that we now predict are causal contributors to Alzheimer's disease. The purple ones among them are the ones that were in genome-wide significant loci. I'm going to take questions at the end. The purple ones are in genome-wide significant loci, and then the gray ones are in loci that did not rise to genome-wide significance. And what you can see is that the genome-wide significant ones are only a subset, and there's many, many more loci that I'm predicting. What you can also see is that this gives us the directionality of the effect. Tells us whether increased expression is positively correlated or negatively correlated with disease, which is extremely important therapeutically as you go targeting these regions to alter biology. But again, everything I've told you so far has focused on a single phenotype at the time. But Alzheimer's and any other disease is much more complex. So we basically said, well, what if we combine multiple phenotypes? So in the case of Alzheimer's disease, there's several signatures. The primary ones among them are amyloid beta, associated with neuritic plaques, tau, associated with neurofibrillary tangles, and neuroinflammation. So we basically said, given the rich phenotypic information for our cohort, can we now start associating individual subphenotypes of Alzheimer's disease with the differential methylation regions of different regions of the genome? And what that led to is the partitioning of the genome, of the epigenome, if you wish, into subsets that are associated with specific combinations of these phenotypes. And these different subsets were enriched with promoter regions and enhancer regions active in very different types of cells. So that basically suggests that coordinately in those individuals, there are many processes together acting on Alzheimer's disease and different facets of Alzheimer's disease. And if you look at the gene enrichments associated with these clusters, again, you see a very rich picture, which basically, again, suggests that we should not be thinking of disease as a monolithic entity, but instead understanding the multiple phenotypes associated with this person. So building on this idea, Yue Li in the group has basically started expanding on electronic health records interpretation, looking at multiple phenotypes together and their co-occurrence patterns to then understand meta-phenotypes or combinations of these phenotypes and how they're associated with disease. Specifically, he's looking at lab tests and ICD-9 codes and prescriptions and DRG codes and doctor notes simultaneously using this hierarchical model that allows him to learn the alphabet, the encyclopedia of terms within each data type, and also how these relate across multiple data types. So that allows him to now start clustering individuals into specific modules of phenotypes, so phenotypic meta-phenotype groups, if you wish, and the data type associated with each of those. And you can see here that these actually span across the many different data modalities of the electronic health record. And the other thing that you can do using this approach is start imputing this information systematically. Because you have a sparse clinical record, when somebody will put one ICD-9 code, they're not going to go and check off every single one of them that also applies. They just want to bill you, so they're just going to check one of them. The doctors might simply, scribble something else, which is actually related, but not always in concordance with this ICD-9 code. And in fact, using all of that information jointly, we can actually start imputing the electronic health records of these individuals and even correcting them, saying, hey, we think that this was incorrectly labeled. Because these phenotypic groups are incredibly strong when you look at all of the information together. And now, how do we relate that to the underlying molecular function? The answer here is, again, very similar to what PredictScan does, of using genetic variants together with the phenotypic clusters to now ask, what is the imputed expression pattern for those individuals based on the genetics of these people look like in specific tissues? So we looked at the Genotype Tissue Expression Project, the GTEx project, which has measured expression levels across up to 1,000 individuals, across up to 40 tissues per individual. And then we're able to say, can I impute the expression pattern of each of those tissues and see how our phenotypic clusters, in fact, correlate with these tissues. So in collaboration with Nikos Daskalakis over at McLean Hospital, we basically found that, for post-traumatic stress disorder, one of the strongest associations appears to be with imputed expression in immune cells again, in blood. And that was weird to us. We said, hey, this makes no sense. But when we spoke with him, he says, actually, there's a lot of literature that suggests that immune processes are, in fact, very important post-traumatic stress disorder. Given the same trauma, different people would develop PTSD or not based on, again, a lot of immune processes. But everything I've showed you so far has been at the tissue level. What we'd like to do now is start dissecting things in specific cell types within these tissues. And I mentioned Alzheimer's in particular because the cell types in the human brain are extremely diverse. So in collaboration with Li Hui Tsai, and again, the ROSMAP cohort and David Bennett, we went back into the same individuals, and we now said, can we look at their brains at much higher resolution by carrying out single-cell RNA sequencing of the same samples? And what we found is that if you measure about 2,000 cells per individual across a cohort of 48 individuals shown here, and phenotypically shown here, including severe Alzheimer's, mild Alzheimer's, and non-Alzheimer's disease individuals, what you find is that there's a small number of clusters that in fact distinguish the different cell types. And if you look at the corresponding function of these cell types, what you see is that they're very, very clearly distinguished, and they're grouping together cells from many different individuals, whether they are AD or non-AD individuals. But for some of them, for some of the cell types, you see subclusters that are actually associated with the disease phenotype. So we basically said, hey, maybe we're only finding these for excitatory neurons because they're the most abundant ones. But if we were to first define the cell types and then subcluster within each of the cell types to look for patterns of expression within each cell type, we may be actually able to find much more detailed information. And that's what we did. We basically took these clusters, we defined cell types within each of those, and then we subclustered within each of the cell types. And what we found is that for every one of the major cell types, 2 clusters emerged, one of which was associated with high amyloid, high Braak stages, high cognitive decline and high AD. The other one associated with low disease signatures, suggesting that within each of the major cell types, you basically have a group of cells whose global expression patterns are in fact changing in concordance with the disease phenotypes. What was also striking is that these correlated very strongly with the sex of these individuals. Female individuals had many more of these disease-associated cells than male individuals, suggesting, in fact, that perhaps the disease manifests much more severely at the molecular level in women, even though phenotypically these individuals were matched. So somehow women appear to be more resilient to the disease at the phenotypic level than at the molecular level.
8 minutes more?
We started about 15 minutes late, so...
We'll still have to start immediately on time.
Okay, sounds great. So then we asked about early versus late changes in disease, and we found that the early changes are much more cell type specific, whereas the late changes are much more common across the different cell types. And we also found that the number of genes that are differential between men and women was increased dramatically in the presence of disease, suggesting that even if we don't see expression differences in healthy tissues, these expression differences between men and women may be much more exacerbated in disease tissues. The other thing we can do with single-cell RNA sequencing data is actually look at mutations at the single-cell level. And what's really important here is that as the brain develops, you can actually have somatic mutations that then propagate to entire regions of the brain, an entire group of cells. So looking for these somatic alterations, what we basically found is that individuals with higher disease phenotypes in fact showed a higher burden of these somatic mutations, specifically in genes and pathways that are biologically consistent. So these were enriched in physical trafficking, Golgi, intermediate filaments, and so on and so forth, which are all processes that are known to be associated with Alzheimer's disease. We also found that the same mutation was in fact found in multiple cells within these individuals that basically suggest some clonal expansion of some of these more rapidly dividing cells. And then lastly, by measuring accessibility information at the single cell level, we're able to carry out the same type of linking between non-coding regions based on their accessibility differences from cell to cell and genes based on their expression differences from cell to cell, enabling us to now start linking genetic variants shown here in black to target genes that they potentially control, some of which are also supported by EQTL information. Using these single cell profiles, we can now go back to the larger cohort of 600 individuals for whom we have bulk expression data and start deconvolving the bulk expression data into the specific cell types. And we can basically see that the cell type proportion as inferred from this deconvolution in fact strongly correlates with phenotypic differences between these individuals. Higher amyloid individuals show fewer excitatory neurons, but higher cognition individuals show more excitatory neurons, and conversely for astrocytes. We can also start asking how these correlate not only with disease status, but with also age. And you can see the number of neurons is in fact dramatically decreasing as individuals age, and astrocytes are in fact increasing. And we can also find genetic variants that correlate with the proportion of different cell types. In this particular case, the fraction of inhibitory neurons, in fact, is very strongly correlated with genetic variants in the STMEG mono6p gene, a locus that has also been implicated in frontotemporal lobar degeneration, but not with AD, not with Braak stage, and not with beta amyloid. So that suggests a link between, neuronal proportion and, genetic variation. And the mechanism for this is the change in a CTCF binding site causing a change in the conformation and a change in expression of a nearby gene. We can also start partitioning EQTLs into the specific cell types where they act. So putting all this together, we can go back into disease loci and start inferring their function, work that we did in collaboration with Melina Klausnitzer. We can start asking where are these genetic variants acting, what tissues and cell types, what are the potential target genes, and how do we link them? Can we find EQTL support for these genes? Can we find the specific upstream regulatory variants that are changing specific motifs and the upstream regulators? And putting all this together, linking them to the cellular processes, we can now start intervening based on the circuitry of these loci, and then altering the upstream regulators, the downstream target genes, and even using CRISPR-Cas9 to edit the single nucleotide variants that are predicted to be causal, which then lead to dramatic phenotypic consequences for these regions. So editing a single nucleotide, for example, leads to this dramatic 7-fold change in the thermogenesis potential of primary cells from risk individuals by restoring this single nucleotide variant. And similarly strong effects are found in mice. Lastly, we've basically started developing methods for doing this in extremely high throughput. So looking at perturbing using massively parallel reporter assays individual SNPs, differentiating stem cells into different cell types to see where are these assays manifesting themselves, changing individual nucleotides in regulatory motifs, doing high resolution dissection of these regulatory regions by tiling them, in this particular case at 30 base pair resolution, and then recognizing what are the tiles that show the strongest activity, and then tiling things at much higher resolution in order to identify individual nucleotides within these regions that are driving the regulatory activity. In this particular case, we're doing a single experiment to test 7 million fragments of DNA, and then by basically cutting them off from the accessibility experiment and then putting them into plasmids, which can then test systematically using self-transcribing reporters where the construct itself is serving as its own barcode. And then that allows us to now start mapping these GWAS regions to very high resolution in specific nucleotides that are predicted to be affected, and even measuring allelic activity in these regions. We are now using these programmable CRISPR-Cas9 constructs in collaboration with Li Hui Tsai to edit individual genes or promoter regions, to edit individual SNPs, and to also turn on or turn off enhancer regions by recruiting activators or repressors within them.
One minute, Manolis, to change over.
So what I've told you about is how we can use epigenomics to systematically map and characterize the circuitry of the non-coding genome, how we can identify disease-relevant tissues, variants, and regulators, how we can combine genetic, epigenetic, and transcriptional variation in disease, how we can do this across multiple phenotypes, learning metaphenotypes that are much more disease-relevant, how we can, in fact, use single-cell data directly or through imputation to deconvolve the bulk activity into individual cell types, how we can use this information to uncover and manipulate specific circuits for disease, and how we can, in fact, do this in very high throughput. This is a set of incredible collaborations with people that I tried to acknowledge along the way, and some of them are sitting in this room, and I'll be happy to stop there.